research. And in particular, I would like to cite BLAST, that most of students in the life science area know. Together with Udi Mamber, he invented the suffix arrays, which enabled the very famous Burrows-Wheeler transform that today is used for space-efficient indices in the context of genomics data. Together with Jim Weber, another colleague of him, was the first to propose the paired end sequencing, whole genome shotgun sequencing of the human genome, the paradigm which most genomes and sequenced are today. Okay, from the period of 2005 to 2018, he focused on the construction of novel microscopes and software for building single-cell expression atlas across developmental epochs. Most recently, he's been captivated by the goal of perfect genome reconstruction using technology to sequence every living species of life. Among the many awards that I can cite, I would like to cite the ACM Kanellakis Prize, which was given to him in 2002. And this prize is given to researchers who have made specific theoretical accomplishments that have had significant and demonstrable effect on the practice of computing. So it's a very important prize. In 2001, he was voted as the most influential bioinformatician by Genome Technology magazine, and was elected to the National Academy of Engineering in 2003. In 2004, he won the International Max Planck Research Prize. And in 2019, so more recently, he received the Royal Society Milner Award for his development of computational techniques that have brought genome sequencing into everyday use, underpinned key biological sequencing tool, and made large-scale analysis of biological images practical. So last, in 2002, together with his early collaborator Webb Miller, he is being awarded to the IEEE Francis Allen Medal in 2022 for pioneer contribution to sequence analysis algorithm and their application to biosequence search, genome sequencing, and comparative genomics analysis. So this is only a small part of his incredible curriculum. So for me, it's a very great honor to have Professor Myers here. We are enjoying his course during this week. So now I give the floor to Rocco, which briefly introduces the topic of the lecture, and then we can start the talk.
Good evening to everybody. Thanks, Professor Pulvirenti, for giving me the floor and to have introduced our special guest. It's a great pleasure for me to introduce the topic that will be discussed this evening, this afternoon, by our speaker, Professor Eugene Myers. Thank you, Professor. I briefly introduce myself. I'm Rocco Di Mauro. I am a software engineer student, a fourth-year software engineer student, and a student of the Scuola Superiore di Catania. And today, I'm glad to represent the whole Corp Allievi during this introduction. And I approached the bioinformatics branch only during last year. So it's a great pleasure, a great honor, and a great start for me to introduce an event like this one. Moving to the topic, to be able to perform haplotype reconstruction is required the computational assembly of shorter haplotype fragments. The reconstruction can be more challenging because of limited fragment lengths and the high haplotype variability across genomes. Reconstruction efficiency represents a turning point for modern medicine and for understanding the details of the evolutionary process. Professor Myers today is going to talk to us about the advances in the technology for haplotype estimation and chromosome level reconstruction, thanks to the new long read and chromosome scale sequencing technologies, which could potentially, I think, change everything on the efficiency of currently used algorithms. Considering his long experience, I am sure he will fulfill our curiosity with several examples of the current state of the art of these technologies, and with a few words of what the future holds for us. And I would like to add that as Professor Myers himself told us during our lecture here in Scuola Superiore, he's a computer scientist, bioinformatician, but moreover, he's a software engineer like me. So I want to, I'd like to hear from you your engineer's opinion on what on the argument that you will treat this evening. Thank you.
Thank you. Can I go?
Yeah, but this one doesn't, I can take this one off. Okay. Oh, okay. So I can use this one. Thank you, Rocco. That was a very nice introduction, and also Alfredo. So it's a pleasure to be here. I've been giving a couple of lectures, and I've been very impressed with the students here. So you have a great school, and I wish you all luck in your careers and in your future. I'm at the end of mine, right? I just retired last year, so I hope it'll be exciting for you. So, I mean, I'm going to give a talk, which is basically going to kind of cover, I'm going to go back to the beginning 40 years ago and do a kind of a brief overview of the history of DNA sequencing technology. So given a DNA sample from an animal or a human being, how do you determine their genetic sequence? Okay. And basically, the conclusion is that we are the thing that's kind of important for you is that we are about to enter into an era where you will be able to take any kind of animal or plant that's interesting to you, and I will be able to give you perfectly its genome sequence. And I mean perfectly, the entire thing. And this is going to completely change science. So for any of you that are involved in the sciences, this will have a very big impact on you. So, but how did we get there? So I want to tell you a little bit, because this has been my journey. Okay, so I started at the beginning, and now I'm here. Okay, and I'm basically going to pass the baton to you because now it's your turn. So, but I tell you how we got here. Okay, so basically, 40 years ago, that was when we basically first started to sequence DNA, and we used a technique called shotgun sequencing. And what's very fascinating to me is that this problem is still the problem today, and still the problem that we face. So in other words, the technique has been the same for 40 years, which is really unusual. Usually problems don't last that long. And so the idea is you'd like to know what the sequence of some strand of DNA. Now, most of you know that DNA is double-stranded. So actually, you read off the code in one direction, and you can read off the code in the other direction. So in my picture, I've shown it as a green strand and a yellow strand in a helical structure. And back in the day, 40 years ago, the piece of DNA that we were trying to determine was typically about 150,000 nucleotides long, right? So DNA is a long polymer over adenosine, cytosine, thymine, and guanine, ACGT, okay? It's a long chain of those. And so to know what the molecule is, it suffices to know what the sequence is, what the ACGT is. So you're trying to, conceptually, you're trying to determine a long string of letters or word of letters, 150,000 letters long of A's, G's, and T's. And the technology at the time, we've never had a technology that will start at one end and read off to the other end that 150,000 base pairs for you. We've had various devices, the technology has been continually changing that allows you to sequence some DNA, but typically only a small amount. Back in 1980, we could sequence a piece of DNA that was 300 letters long, just 300. Today, we have technologies that do 10,000, but 10,000, not 150,000, or, to do the human genome, it would have to be 3 billion, okay? So the problem then is so given that you can't literally directly read this off, what we do is we randomly sample the sequence. So we take many copies of it, we shatter it into pieces, and we run those pieces in the sequencing instrument so that we get many samples of this overall thing. We don't get the whole thing, but it's like we're getting little pieces of 500 base pairs. It's like we're reaching in and saying, here's another 500 that's in there. Here's another, okay? And So, and we call those reads. And back at the beginning, they were about 500 letters long. Okay. And we would collect enough of these, okay, that you would basically be able to reconstruct the target. So you have to collect a lot of data. So we would collect, for example, for this 150,000 letter target, we would collect about 1.5 million letters in 2,000 of these reads. So I wouldn't produce just enough reads to cover it once. I would go in and I would produce enough of these reads so that it's as if I had sequenced it 10 times. So I produced 1.5 million letters of data in 2,000 reads. So I basically oversampled it. We call that the coverage, and we covered it 10 times. Okay, and the idea was is that you need to do that to oversample it to basically be able to reconstruct it. So the problem, and again, this is the problem that we've been facing for 40 years, is to build what's called an assembler, which is a classic inverse piece of software. In other words, it says, okay, I had this thing, I probed it 2,000 times, give you some information, 2,000 probes. What was it? Okay, what was it? Okay, and so what and the problem is made more difficult. It's because there are a number of problems with the sequence. First of all, there are errors in the reads. The reads aren't perfect. There's like 2 to 5% error. So in a 500 base pair read, about 10 to 15 to 20 of them would be wrong. Okay, so I drew those as little red dots. Okay, to indicate those were errors. And then the other thing that you don't know is you don't know whether the read came from the green one or the yellow one, right? You don't know which strand it came from. So they're running in different directions. Another problem you have is that there are various readout artifacts. Sometimes the pieces anneal together, so you get a chimer, or some of the artificial DNA called a promoter is present, or sometimes you were a little sloppy in the lab and you had not only some human DNA, but you also had some E. coli or bacteria in there. Okay, so you have contaminants. So those are additional problems. And then another problem that's intrinsic to the combinatorics is that the genome is repetitive. There are parts, oftentimes in DNA, there are parts where one part is almost exactly like another part. Okay, it's repeated. There's 10,000 letters, and you see almost exactly the same sequence of 10,000 letters elsewhere, and that compounds the problem of inferring what this thing is. And then the last thing is, even though you sampled this thing 10 times over, there's still a small chance that you didn't sample all of it, okay, probabilistically. But the problem of the assembler is to basically take this data and to reconstruct, to basically put together the overlapping reads in a way that it's convinced or correct. Okay, so to basically produce what are called contigs, because they're contiguous stretches. Okay, and the idea is that the assembler is supposed to be conservative, and if it can't quite figure something out, it shouldn't put it together. And in addition, because you've oversampled it, you can actually also vote out the errors. In other words, if you sampled it 10 times over, you expect a pile of about 10. This is just a cartoon, and you can basically then vote to basically fix up the errors and produce a better consensus of what the original thing was. Okay. All right. And again, because either the data didn't cover the target, or because the assembler just couldn't quite figure out this part, because maybe it was too repetitive or too complex for it, you get gaps. Okay, so you don't necessarily get a contiguous sequence out. So in the very first shotgun project was performed by this guy, Fred Sanger, and his team in England, and they sequenced an entire phage. They sequenced a little virus called Lambda. It was about 48,000 letters long, and they did it with exactly the method that I'm showing you. And they were the first ones to do it. In fact, this is their method. Okay, and I'm pointing at this paper because if you're a student of history, this is, I think, a very important paper to read, because it was an incredible paper. It was just 5 pages long. And in those 5 pages, they introduced 3 techniques for actually doing the sampling, for producing a random collection of reads. They produced 4 techniques for trying to fill the gaps. And they even presented a simple rudimentary program for actually trying to automatically put all the data together. It was an incredible paper. in a single paper. And, you know, you know, so this is, in essence, this is a paper whose standard we should all aspire to. Okay. All right. It's an example of an excellent scientific paper. And it also explains why this gentleman won 2 Nobel Prizes. Okay. Not just one, but twice. Okay. So in this, so what I'm going to do is I'm going to basically kind of fill out a table in which I'm going to show you how the technology advanced. Okay. And at some point, I'm going quite slowly because I think this is a general audience, right? Usually I don't explain quite so much, but I'm going to go slow and then I'll skip over some things. But I'm going to basically fill out a big table where basically we're going to talk about how big is the target, that's the genome, 150,000. How big are the reads? They were 300 to 700 letters long in the initial technology, the Sanger technology, what the error rate in those reads was, and how much you had to cover the genome, 10x in the time. So in the Sanger era, which went from 1981 for almost 20 years to 2005, this was kind of the standard that decides the problem. So what happened next wasn't so much that there was really a big change in the technology. In fact, the technology didn't actually, it was kind of depressing. The technology didn't advance that much. What improved was there was some better automation for producing the data, and they were better at kind of handling and manipulating the samples. And what happened next is using this really the same technology in a slightly different way, as you'll see. What happened is the target, instead of trying to just sequence 150,000, the goal became the entire genome, okay, which in the case of humans is 3 billion, okay? So if you go back to 1998, okay, if you went back, and it's probably, I don't know, were you guys born yet? I don't know. It's the belief was that you know, the 150,000 number that I gave you was the biggest thing you could sequence. You know, it was a 2,000 piece jigsaw puzzle, 2,000 reads. Okay. You had to kind of figure out how to put them together. 2,000 piece jigsaw puzzle, right? You guys have done 1,000 piece jigsaw puzzles, right? Humanly, you know, you can do one. Okay. And so that this was about the biggest thing that you could do. And so in 1998, the public sector project, the Human Genome Project, that was run by in the academic circles. The idea was, well, look, I want to sequence human chromosome 1, which I'm depicting by this line, but human chromosome 1 is 150 million letters long. Okay, so it's 1,000 times bigger. Okay, and the question was, well, how the heck am I going to do that? Okay, and so what they did is they basically shotgunned the DNA, okay, into pieces, into many pieces, but these were big pieces. They were 150,000 base pair pieces. And they had figured out a way called a back artificial chromosome to manipulate those and keep those, okay, and to actually infer some information about them. So the plan was basically to do kind of a meta level shotgun assembly. So they were going to shotgun chromosome 1 into big pieces that they could manipulate in back arterial chromosomes, 150,000 base pair pieces. They were going to figure out how those really big pieces went together into what was called a physical map. Okay, I won't explain the terminology there. And then the idea was once they had the physical map, they could then pick kind of a minimum tiling of these clones of these big pieces that covered the chromosome, and then they would take those and shotgun sequence them. Okay, because they're 150,000 base pairs, I can shotgun sequence them. Okay, and this plan had a very good characteristic, which is that it was guaranteed to deliver something. Okay, and you know they were using taxpayer money. Okay, your money. Okay, so they wanted to make sure they gave you something, and that's good. Okay, so it was a secure plan. It was definitely going to produce something. It might not have produced a contiguous genome, but it was going to produce a lot of the genome in big chunks. It was secure, but it had some problems. One was that as an industrial process, you had to have a factory for doing shotgun sequencing, and the technology for manipulating and managing these pieces and assembling them and collecting the data about them was completely different. So you had to have 2 separate factories, okay, not just one. The other problem is that the ability to keep these pieces was not stable. These clones, you would take a back 150,000 piece, okay, you would put it in the freezer, and if you pulled it out 6 months later, it was not the same piece of DNA. It oftentimes it modified. So 4%, oftentimes after 6 months, 4% of the clones weren't exactly what they were before. Okay, so that's a problem. Okay, they had rearranged themselves. The clones were unstable. The other thing is the information that you could collect about these to figure out how they went together was much less precise than the sequence. And so these maps were very difficult to build. In fact, they never built a complete map of the human genome. Kind of a little dirty secret. They never actually finished this phase. Okay, and then finally, the other problem is that there are going to be like 20,000 backs that need to be shotgun sequenced. So you have to have a technician. You have to have an army of technicians doing 20,000 shotgun protocols, okay, which is a huge amount of effort, okay. So, so there were some interesting things that happened around the same time. In 1985, Craig Venter, at his institute, Tiger, basically sequenced a 1.8 million bacteria called H. influenza. And he did it with shotgun sequencing and then a process for gap closing. It wasn't quite a whole genome shotgun, but what that event did is it made everybody think about, well, wait a minute, at least it made me think about, is 150,000 really the biggest thing you can do? Because look, Craig just did 1.8 million base pairs, okay? Which is, not supposed to be possible, right? But yeah, no, it is. Now, admittedly, the bacteria has a very simple sequence, okay? But still, it's like, So I got together with this guy, Jim Weber. Oh, and I can't go backwards, can I? Can I go backwards? Does it? Okay, so Jim Weber and I basically, based on kind of this inspiration, we kind of re-looked at the thing and we proposed basically a paired end protocol, which I'll describe in a moment, that basically, and that we could then shotgun sequence the human genome. In other words, instead of doing 50,000, 1,000 piece puzzles, we were going to do one 50 million piece puzzle, right? So it's a pretty big jigsaw puzzle, right? 50 million pieces, yeah? Okay, but I won't belabor it completely, but we had reasons to believe that it was possible, and we put forward, I think, a good argument for it. And the idea was to do paired end shotgun sequencing. So what's different in paired end shotgun sequencing is you fragment the genome again into pieces, but this time the pieces are a bit bigger, and you carefully size select them so you know how big they are. So you run them through a gel and you select them, the ones that move a certain part in the gel, those are all like roughly, 5,000 base pairs long. You take those, you cut those out of the gel so you can precisely size the pieces now. So I only take the pieces that I get that are like 5,000 base pairs. So I know the size of pieces. And then what you can do is you can arrange a sequencing reaction at both ends. So you're still only getting like 500 letters. Okay, you're only getting 500 letters in a shot, but you're getting it in pairs, right? Because you're sequencing both ends. So you're getting 500 of one end and 500 at the other end. You can't get any more than 500, but you can at least, so you get pairs of reads that are at a certain distance apart, right? They're five because you carefully size the DNA. So they're 5,000 letters apart. Okay, and so everything's as before in terms of the caveats and the problem, except now the assembler can do something a little bit extra, which is because it knows the pairing relationships. For example, if it has a pair in this contig and another pair in this contig, and it knows the distance, then it has an idea that this contig is before this contig, and it even has an idea of how far apart they are, right? Because it knows how far this distance is. So you can basically take your contigs and you can arrange them into linked scaffolds, right? Where the pairs basically show you how to connect the dots because the pairs span the gaps, okay? They span any gaps in your problem. So basically in 1998, Celera was actually formed to do exactly this. Okay, so this was very exciting for me because I didn't know that PerkinElmer was going to do this, but they formed this company, and I immediately called Craig and got on the team. And the idea was is that we were going to do pairs, but we weren't just going to do one size. We were going to do 12 million reads in pairs that were 2 kb apart, 2,000 letters apart, 12 million reads that were 10,000 letters apart, and 2 million reads that were 100,000 letters apart. So we were getting kind of a spectrum of paired distances. And this was kind of the reaction. Okay, this was actually from the New York Times. Okay, and this is what they thought of our approach, right? So they didn't think we were going to have much luck with the 3 billion pieces. Is it's not 3 billion, it was 50 million. Okay, a smaller number, but they kind of everybody said it wouldn't work. Okay, which actually is very exciting because I'd been studying it very carefully and I had a feeling it was going to work. Okay, and it was kind of very exciting for me because when I asked people, well, why isn't it going to work? Okay, I never got rational answers. I always got emotional answers. Okay, which to me was a cue. Okay, that people just didn't want it to be true. Okay, they just didn't want it to be so. Okay, and so to me that was very exciting. It was like, wow. Okay, this could be interesting. Okay, all right. All right. So, and just to show you, I mean, how I actually did it, it turned out that the secret, okay, the trick, which in hindsight is that hard, is that I had to figure out a way to understand what was repetitive versus what wasn't repetitive, okay? Because the parts of the genome where there aren't, there's just one copy of it in its unique sequence are actually very firm and easy to see. And the question was, could I identify that versus the repetitive stuff? And so I won't belabor this because it's not a technical audience, but I figured out a way as a computer scientist to do this. Okay. And that was the important thing. And so what I would get is that I would be able to put together. So these are all the pieces that I knew went together in one way and didn't represent something that was repeated in the genome. Okay. And I found a way to do that for the geeks. I found a maximal uncontested interval subgraphs that had the right coverage. Okay. Right. So, but there were like 300,000 of these. Okay. And what order are they in? So if we had stopped there, they would have been right? It would have been Swiss cheese. I would have said, oh, here's the human genome, 300,000 pieces. And by the way, I have no idea what their order is. Okay. But remember, I have the pairs. Okay. So with the pairs, right, I was then able to link them together. And with the pairs, I was able to link the pieces into scaffolds that were that spanned millions of base pairs. In other words, were very coherent. So instead, I was giving you the genome in about 1,000, 2,000 big pieces, which for the science at the time was considered very contiguous. Okay. And it was by basically finding pairs of linked reads that, pulled everything together and pulled it in a scaffold. But you could do even more than that, right? Because these holes represent the repetitive portion of the genome, and even the repeats could be solved by basically doing what I call throwing stones, which is if I had a paired read where one of the reads was in, say, in this contig, and by knowing the distance, the other read, I knew it had to be in this hole, right, between these two contigs, then I know that this read goes here. And so I basically looked at all of the read pairs where the read was in here, or one of the reads was in here, and the distance said that the read had to be in the middle. And then I took those and I assembled them because in isolation, that's unique, right? It's only a repeat with respect to some other part, right? But I've got the things that are right there. So I was able to backfill most of the repeats, right, to give you a very contiguous sequence. So what was cool is in 2000, this was a 20,000 CPU compute, okay, hour compute, which was a very big compute back then, okay. We managed to do it in a 64 gigabyte memory, which was incredibly expensive back then, okay. I mean, you guys know that at one point they were suggesting we buy a terabyte of memory and it was going to cost $1 million. Okay. We didn't do that. We did a 64 gigabyte. I saved the company a lot of money. They loved me. Okay. Anyway, so as a computer science, but and what was very pleasing to me is that, well, and then basically and to prove the efficiency of this, I mean, we started in 1998 in an empty building. Okay. There were 8 employees. Okay. you know, by 2001, the company had 1,000 people, okay, and we had sequenced basically not only the human genome, but the fruit fly, the mouse, and the mosquito, okay. So in other words, you know, because the Human Genome Project was supposed to finish in like 2008, okay, and so this accelerated the whole thing, right? And not only did we sequence the human genome in less than 3 years, we sequenced all these other genomes, okay, which proved kind of the efficiency of the method. And, you know, to me, it was very exciting because it, you know, bioinformatics is, you know, especially at that early stage, it wasn't quite clear, right, whether, you know, guys like me had any value to the biology or to the genomics, right? And so this was a clear win, right, for the geeks. Okay. All right. So I was very happy about that. All right. So what happened next, okay, is that basically in 2004, we started the era of what's called the short read era, where again, the target was the genome, but the reads now, it's a different technology, and I'll show you the machines in a minute, but the machines produced about 150 letters, so even shorter than Sanger reads, but they were much cleaner. They only had a 0.5% error rate. And they were so efficient that you could produce tons of data. So people increased the coverage. They would do 30-fold over sequencing to try to compensate for the shortness of the reads. Okay. And so these are the instruments. And basically you can see they you know, the output was increasing. By the end of it, Celera had 300 machines. Okay. Today, one of these NovaSeq 6000s, okay, is basically equal to like 10,000 Celeras. Okay. It's 36 Celeras. It's equal to 10,000 of the machines we had at the time. And they were producing tons of data, and it was kind of a very interesting time. But to me, it was a very depressing time because the goal, okay, of these short was basically to was the mantra at the time was the $1,000 genome. Okay, so the NIH comes out 2003 and says the goal is the $1,000 genome. Okay, but so people were working on methods that would produce a lot of data. Okay, but they were producing these really short reads, 150 base pairs, and all you could really do with those reads was what I call resequencing. So if you have the human genome, so you have one human being's sequence, right? So we had one human being's sequence, okay? At least Celera did. The public sector was actually a chimer of 5 people. But the point of the matter is that our genomes are very similar. They're like 0.1% different. Okay, I mean, really, there's not a lot of difference between us. Okay, you know, and except for that pesky Y chromosome, and so basically, what you could do is you could take those short reads and you could map them. You could find out where they were in the human genome. But if you tried to assemble them, it was crap. Okay, you couldn't figure out how the pieces went together. And in fact, I'll show you that by this example. So here was when we did the fruit fly, Drosophila melanogaster, the average kind of size of a contig was 20 million base pairs. Okay, so we had most of it in big, you know, chromosome-sized pieces. A little bit later, somebody did Sanger reads, so those 500 base pair reads, but without the pairs, they got 180,000. Okay, and then even later, somebody just did the short reads, the 150 base pair reads, but they did them in pairs. They got 20,000. Okay, and then somebody even later, fly number 3, did 100x of this next generation sequencer, 550 base pair reads. They got 8,000. They got contigs of 8,000. Okay. All right. I mean, that's 1,000-fold difference in continuity, right? 3,000-fold difference in continuity. And this assembly really was just a whole bunch of collections of small pieces. It was crap. Okay. It was crap. Okay. And to me, and I knew it was going to be crap. Okay. Because then, you know, the mathematics would tell you immediately when the reads are that short and unpaired that you just don't have enough leverage to try to figure out the sequence. Okay. And so it was in, but for mapping, if you find a, you know, 150 base pairs and it goes there, that's fine for mapping. So you could basically resequence people. And for the doctors and the medicine, this was great because then we could, you know, we could go sequence you, okay, cheaply on one of these big machines, and we would be able to see how you were different from the first person, right? And then we map your sequence and your sequence and your sequence, and then we can start seeing how there's variation in the human population, right? So for medicine, it was good because we could sequence people over and over again, okay? But I swing more as a scientist. Okay. I like the scientist more. I want to go out there and explore the natural world, right? So I see some beetle, okay, or some weird bat. I want to know its genomic sequence because I want to know why that bat's so weird, right? Why can that bat eat sugar all day long and not get diabetes, right? I mean, those are the kind of questions that interest me, right? But you can't do that with this technology, right? You can't do what's called de novo sequencing, right? Or ab initio sequencing. You can't without a model. You have to basically have already a sequence determined in some other way, and then you're mapping to it. So it's called resequencing, okay? And the genomes were crappy, okay? So fortunately, okay, things changed. In 2012, I actually left the field. You know, in 2005, I wrote my last paper, okay, about genomics, and I said, okay, I'm done. Okay, these guys can go ahead, okay, and resequence everything they want, but you know, there's no interesting problem left. But fortunately, in 2012, okay, the third generation of technology started to come out, the so-called long read sequencers, okay, and these are pretty interesting devices because they produce reads that are 10,000 to 35,000 base pairs long. So the reads are really long. Yeah, they're much longer than the other ones. But the error rate is 10 to 15%. Okay, really high error rate. Okay, but again, inexpensive enough that I can produce a lot of data. I can produce 60x. And so I don't want to spend too much time on this because I'm probably how much longer should I go? Are you guys interested? Is it good? Is it good for you? Are you following? Yeah, because your general audience, right? You guys aren't all like, you know, informatics, right? You know, okay, you don't all write computer code, right, all day long, right? So some of you do interesting things. Okay, so I'll try to finish up in like 10 minutes. Okay, all right. So huh? Yeah. Okay. Well, let me know. I mean, I'll try to gauge whether or not you're still interested. But basically, what's interesting about these machines. So here was the first one of these machines. They were really expensive. This thing cost almost $1 million. Okay, that was one thing, and it was a boat anchor. Weighed like, I don't know, weighed like a ton. You know, it was a cow basically. But it was kind of cool because what it was doing is it was actually this generation. These were single molecule machines. So what the machine was really doing was it was actually following the construction of the complementary strand of DNA in a zero mode waveguide. So in other words, it's like a 250 nanometer little hole that you basically figure out a way to get the DNA kind of anchored at the bottom with biotin, and then you have like labeled nucleotides, and you watch their incorporation. So you're literally like watching the molecular process of DNA duplication. Okay, except instrumented. So biotechnology. So very cool. And the problem though is that, you know, there's very little illumination. So there was a huge amount of noise, thermodynamic noise, and it was hard to see these things. It was kind of at the limit of what cameras could do in 2012. But there was a couple of interesting things, which is first of all, the errors that it was making the sequence, yeah, it was 10%. Okay, every 10th letter was wrong. Okay, but the noise was completely random. In other words, it was a result of a thermodynamic process, which is another way you make actually a random number generator, right? So when an error was happening, it was completely mixed. Okay. The other thing that was kind of cool was because it was single molecule, you know, when you sheared all those pieces and you put them into the instrument, every piece with equal likelihood got into a well. So every bit of the genome was being sampled equally. Okay, so the sampling was truly Poisson. And this was not, I don't have time to belabor, but this wasn't true of the previous technologies. Okay, I could bore you with another 10 minutes of how those why those technologies didn't have these properties. But so I basically got on the phone with the CEO of the company, Mike Hunkapiller, who's a guy I worked with at Solera, by the way. So it's, you know, and he said random in this phone conversation when he was pitching the machine. He said the word random, and I realized, you know what? That 10% is not a problem, because if the errors and the sampling are random, then if you produce enough data, you can outvote the errors. You can basically come to a perfect consensus, right? The only reason you couldn't come to a perfect consensus with the other instruments is because error wasn't random and because the coveraging wasn't complete. But now I have an instrument that will cover the genome reliably, cover it to any depth I want. And if I go deep enough, I can vote and get perfect. So I realized as a mathematician that this machine had the potential to produce fantastic genomes, okay, of very high quality. So in fact, I got so excited, my first tweet was a theorem, okay, that this was true, this was possible, okay. So I basically, I had just moved to Germany. I was a Max Planck director. I had a lot of money in my back pocket, okay, because I was a Max Planck director. That's a good thing in Germany. And so when I got there in 2013, I said, man, I'm going to do this. So we built up a shop with these machines and others. So we built a genomics center, which eventually went on to become a genome center for all of Germany. And I basically embarked on 2 pilot projects, this planarium genome, which had defied all attempts at assembly. It was 80% AT rich, okay? And so it was very hard. And so we got 100x more continuity than any previous assembly. And then I tackled the 32 gigabase axolotl genome, which at the time was the largest thing ever sequenced. So it's 10 times bigger than your genome, okay? This little axolotl, this little salamander, it's got a genome 10 times bigger than yours, okay? And so I was going after a record, and we published those things, and those were really good assemblies. I mean, they weren't at the quality we can do now, but they were compared to what was being done. These were better than the human genome, okay? Let's put it that way, okay? That we produced, 20 years previously. And so I began to realize, like, look, over a period of time, we're going to be able to produce almost perfect high quality telomere to telomere reconstructions, okay, of genomes. We're now embarking on a trajectory that's going to take us to being able to do this perfectly. Because when we did the human genome, when we reported the human genome in 2003 and we had the big press conference and we went, oh yeah, the human genome sequence, we'd actually only put together about 90% of it, okay? Okay, today we can put together every single bit of it, okay? Perfectly, okay, which is pretty, I mean, that was actually just happened last year, okay? And it's because of these technologies, okay? And so, I got involved. There were a few other people like, guy Eric Jarvis at Rockefeller and Richard Durbin at the Sanger Center in England, that we all kind of saw this potential around 2015. And so we got together and we created some projects whose goal is to go out there and sequence the natural world. So we're going to go out there and sequence all the vertebrates. There are about 70,000 vertebrates. We're going to go sequence all the bats. There are about 1,400 bats, okay? And so, for me, it was a conduit for kind of going back to work on the assembly problem, okay, which I got very excited about. So I think I'm going to, so basically I need to cut it short. I mean, basically with this new technology, we could produce really beautiful assemblies. We could basically get, a Contig N50 that was in the mega base pairs, millions of base pairs was the average contiguous piece before you hit a gap. And we could put it in and we could then scaffold to the point where the scaffolds were 40 to 50 million base pairs. I mean, basically in 2017, 2018, with that technology, we were able to basically produce an assembly of a genome in which each chromosome arm was a single scaffold, okay, with a few gaps in it, okay, which is really cool. I at one point I did a calculation and I showed that basically if we tried to go out, so if we embarked on a project whose goal was to go out and sequence everything on the planet, okay, in today's dollars with today's technology, it would not cost more than the Human Genome Project, okay? So it's not a crazy idea to think about going out there and sequencing everything on the planet, okay, with this technology. And I thought in 2015, I thought we were ready to put on the show, okay, but I was actually a little bit early because in 2018, a slight refinement of this technique changed the whole equation because the reads were now, it was basically using the same technology, but with a slight change in the protocol in what you did in the machine. And so we were getting reads that were 15 to 20,000 base pairs long, so still very, very long, but with an error rate of 0.2%, okay? All right. And in fact, with some finagling, you can get down to 0.01%. So we're getting almost near perfect reads. Okay. And we would do it at 30x. And I'm not going to belabor how you get those. Okay. I'll just say that, okay, because I want to kind of fast forward. But with these almost perfect reads, okay, we're now able to produce genomes where the typical genome is the contig N50 is 10 to 15 megabase pairs. So another order of magnitude improvement, right, in continuity. 10 million, no, not a million, I'm 10 million. And we would get 100, 200 big pieces. And basically the typical scaffold would be a chromosome. Okay, so we were basically, it was complete. So if you think about it, at this point, we've got genomes that are end to end, telomere to telomere. Okay, and they have, but they have a few gaps in them, not many, 100, 200 gaps, and not very big. Okay, so almost. Okay, and with a lot of extra effort, and if you collect data and you're willing to spend more money, you can make them perfect. And Philippi showed that a year ago. Okay, so so this vertebrate project and all these other projects, it's now spawned this idea of biodiversity genomics. So we're finally at the present. Okay, and so 5 more minutes and then I'll stop. Okay, but I want to tell you what's happening now. Okay, so these projects, it's like there's a lot of people now who get the idea, right, and understand that with these technologies, we're going to be able to go out and, you look at some interesting bat that lives 50 times longer than it's supposed to, 50 times longer than it's supposed to for the function of its body weight. You can go out and sequence that, see what's going on inside of it that makes it, live so long. Okay. And, and so now everybody has that idea, right? So the topic has become called biodiversity genomics, like really to go out there and sequence all the flora and fauna on the planet to see what's going on, kind of at a molecular level. And so there's all these projects. I talked about the vertebrate and the bat project. There's now a project to sequence everything, the Earth Biogenome Project. There's another project in England which is focused on just sequencing everything that it's in the British Isles, and another one in Europe to do just all the things in Europe. Okay, so there is one called ERGA. There are a number of Italian universities that are involved in this project. And of course I've been involved, and this is when I give this talk to my peers, I want to brag about how much data I've produced. Okay, so those are the numbers that give my data. Okay, but so I've sequenced a lot of bats. I love bats. Okay, so no, bats are really cool. They're we sequence mice because they're close to humans, right? But actually, bats are mice with wings that are much more interesting, okay, because they're extremophiles. There are bats, like I said, that live 50 times longer than they're supposed to, okay, as a function of their body weight. You'll find another, sister species, 5 million years diverged, that lives a year, and then there's this species, 5 million years diverged, lives 50 years. Okay, you can find bats that, like I said, there's one that eats fruit, that eats basically sugar all day long, never gets diabetes. Okay, there's another one that there are other ones that basically never get sick, perfect immune systems. They carry Ebola and all these nasty things, right? Okay, they don't get sick. Okay, they carry SARS, they don't get it. Okay, perfect immune systems, right? So in terms of their characteristics, right, bats are fascinating, and they've got some characteristics I'd really like us to have. I would like to be completely immune to everything. Okay, I would like to live 50 times longer than I'm supposed to. Okay, and I would like to be able to eat torta all day long and never get diabetes, right? So I want to study bats. So I'm studying bats, right? Okay. Now you know why. Okay. So here are some other things that I do, but I want to show you one last thing, which is kind of very interesting. So this is also, one other very subtle thing happened when we got to these very high fidelity reads, because previously, that the DNA is double stranded, right? There was the green one and the yellow one. And in all the reconstructions that we were producing, we basically were giving you the consensus. In other words, the average of the green and we weren't distinguishing them, right? But you got one set of chromosomes from mom and another one from dad, and they're not exactly the same, okay? And with this new technology, what we're able to do is we're actually able to separate the two haplotypes. In other words, instead of giving you 22 chromosomes for a human genome, I'm going to give you 24. I'm going to give you 48. Okay. I'm going to give you the set from mom and I'm going to give you the set from dad. And it's the first time that we've ever been able to do that. Okay. And in fact, I was we were at Okinawa with my colleagues Takeuchi and Sato. We actually reconstructed the pearl oyster, okay, in this way. We haplotype phased it. And this slide basically just shows you how much it's improved, right? This is what you did with the short reads. This is the long reads. You can see they're getting longer. This is kind of the length of things, and then the hi-fi. So you can see each kind of one of these things that I've been talking about has been rendering an order of magnitude improvement in terms of the continuity. And then, of course, when you do this scaffolding. So so for the pearl oyster, for the first time, we were actually able to separate them, okay? And I got a confession. It's a little bit easier to do for the pearl oyster because the mom and the dad are 1% apart. Your mom and dad are 0.1% apart, right? So it's harder when they're that close. But what was fascinating was when we looked at them, okay? Most of the time, the mom and dad, the paternal and maternal, were basically almost the same. You know, there was a little bit of variation, but they had the same genes, you know, in the same order, you know, coding for the same stuff. But in one chromosome, chromosome 9, we actually found like a few regions that were like millions of base pairs long, where they weren't the same at all. Okay, and in fact, we were so startled by this that we went back and we double checked it and triple checked it to make sure that we hadn't made a mistake. They weren't the same at all. Okay, they had different genes in them, okay, in different orders, okay, which is really weird. And in fact, what's interesting about that too is that, you know, when you make a copy of your chromosomes, right, in order to make the copy, you have to have they have to be similar. And because they're similar, they can also recombine. So you can get cuts and breaks. In those regions that aren't similar, you can't have any recombination. So these regions are completely resistant to recombination, which means that they persist. Okay, in other words, they keep going. Okay, all the pearl oysters have this difference. And guess what we saw when we looked in there? Guess what kind of genes they were? Most of them were genes that were involved in immunity, in the primary immune response. Okay, and now this is all conjecture. This hasn't been verified, but this is our hypothesis. Okay, which is these were involved in the primary immune system, and so by virtue of having two distinct repertoires, it had a very kind of diverse immune system, and this would explain why when the Japanese do horticulture and they crossbreed, okay, and they kind of try to inbreed oysters, they get oysters that aren't healthy because what they're doing is they're getting only one of those in the inbred oyster, right? And so the thing's not healthy and it dies, okay? So we're trying to verify that now, but you see, that might be some very important biology that wasn't apparent, okay, until we were able to separate these things. And in fact, we've now seen it in corals. We now see it in worms. So it doesn't look like it's something that happens in us. It doesn't look like it's a mechanism. But for a lot of lower eukaryotes, this appears to be very interesting biology. Okay, so that's the end of the story. And so like the conclusion basically is that we are very close to being able to go out and sequence things perfectly. It's still a little bit expensive, but eventually it's going to be cheap. Okay, you don't have to be much of a futurist to know that the cost of technologies always go down. Okay, and it's been getting cheaper and cheaper. Right, it was very expensive. That machine was $1 million. You know, the machines are 500,000, but the runs have gone down. It used to cost me 50,000 to do a human genome. Now it costs me 3,000, and we're on our way to like less than 1,000.